To get started, we're going to ask the panel members to introduce themselves. Some of them have already been introduced, but some of you have short memories. So we will ask them to introduce themselves again. And I've also asked them to say a sentence or two about some burning issue that they want to make sure you've heard from them, just in case we don't get to it in the Q&A that follows. So we'll start with Bill.
Hi, everybody. I'm Bill Hait. I'm the global head of R&D at Janssen, the pharmaceutical companies of J&J. There were three things I wanted to perhaps touch on. One was immorbidity. The second is why soon we'll be more like our cars than ourselves, and then get back to behavioral science.
Terrific. Heidi.
My name is Heidi Williams. I'm an economist. I am an assistant professor in the economics department at MIT. And as an economist, it's not surprising that I'm going to try to talk about incentives. And so when we think about what's scientifically feasible, I just want to stress that that's a product of the private research investments that have been made in the past and the public research subsidies that have been put out there. And so I spend a lot of time thinking about how we could change policies in order to better design private investments to be targeted towards the things that are most socially valuable, or use public research dollars in a way that would best benefit patients.
Doug.
I'm Doug Lowy. I'm the acting director of the National Cancer Institute. And what I would like to tell you is, first, there's an enormous amount that we have learned over the last few years about cancer, but there still are enormous gaps. Therefore, we need to do a lot of research on that. To try to improve the outcome for patients who get cancer and prevent and screen for cancer, we need to think seriously about how we can take new information and apply technology to solve those important problems. And third, there are enormous challenges, but also enormous opportunities for applying the technology and the new understanding in the developing world, where the need is actually greatest for trying to bend that curve for cancer incidence and mortality.
Bert.
I'm Bert Vogelstein. A message that I would like to give to all the brilliant young people in this audience is that if you're interested in making a difference in cancer, please at least think about opportunities in prevention and early detection rather than focusing your effort on trying to cure advanced cancer patients.
Saengeeta.
I'm Saengeeta. I just spoke. But I actually echo my panelists' comments. The two things I wanted to touch on were how to get some of these early detection technologies, how to accelerate their movement into the clinic. And there are lots of structural, I think, disincentives to do that. And the second thing is to sort of shine a light on the global oncology need. 70% of cancers will be in the developing world by 2020. And this is a setting where there's no screening, there's no radiation, there's not steady electricity. So, I think for the trainees out there, this is something really to think about. Maybe the questions are actually different than the ones that we've been asking.
Ed.
So, I'm Ed Scolnick. I talked about my urine, and I guess I would have a couple of different messages. I think that the problems need to be identified with respect to each kind of cancer that we'd like to do something about. And if you use lung cancer as an example, whether there needs to be one approach or multiple approaches is really an important public health problem. That is, should the focus be on early detection, where, as Saengeeta pointed out, as opposed to the talk that was given earlier, the current early screening methods don't seem to work very effectively. So we know what the risk is due to and what has elevated the risk. So obviously, we must need better early screening methods than we currently have in order to move the needle. And it's, in addition, a public health problem. And I think that at least this country, and probably no country, has really tried to develop acceptable ways of incentivizing people to take better care of themselves so that the risk goes down. So I think that's one important message, and I think that's probably going to be perhaps different for each cancer because the different causes of the cancers, of different cancers, are different. The other point I would make, picking up on, I have to do this because Doug talked about the HPV vaccine, and I was involved heavily in making the first one. All the pathophysiology that Doug talked about, that his group so elegantly worked out, we had to guess would be true at the start of our project. And that's the argument for what Doug has just talked about. That is, if you want the commercial world, whether it's a pharmaceutical company treatment or a diagnostic test, to improve the lives of people, you have to build that on the basic research knowledge that is mostly fundable through NIH and sometimes through private donations. And there is absolutely no substitute for that, whether it's in this field or in other fields. And if that information is not available, and I can tell you in the HPV field, the amount of information that was available that would have predicted the project would work when we committed huge resources at Merck to make the vaccine was minuscule. It was really minuscule. And I think that Doug deserves a lot of credit for what he's done over the years, but without the basic research enterprise, companies will not try to implement the tests or the preventions or the treatments that really will move the needle.
Yeah, we certainly agree that basic research is the foundation of everything. So we know that for all progress, we need to continue to invest in basic research. Many important questions still to be answered. So I will touch on some of these things in my questions to you, and I encourage, again, the panelists to interrupt each other, argue with each other, fight one another. Please don't get out of your seats, but we want to engage the group. And I thought I would talk about really two areas in my introductory questions to the group. Firstly, in the area of early detection, and then we'll come to prevention and other aspects of early detection and some of the implementation and system-wide challenges that exist that maybe are limiting our progress in these areas. But I want to start with early detection, and I want to kind of take up the challenge that Barry Kramer raised, which I think is an extremely important one. When Barry was talking, I was actually thinking about that movie, Kramer versus Kramer. Remember that movie? I recast it now as Kramer versus everybody else. So, but Barry makes very important points about the challenges in population-wide screening with tests that have limitations with respect to sensitivity and selectivity. And I want to focus it on an aspect that he described, that is to say, distinguishing those lesions that are detected that would have done nothing to you over your lifetime from those that would have killed you. Because I think if we can solve that problem scientifically and technologically and implement it properly, we'll meet Barry's challenge. And I talked to him at the break, and he agrees with me. So Saengeeta, you talked about this with your new technology. Can you amplify a little bit, and then others can join in as well?
Sure. I can tell you how we're thinking about it. I think the end of the story, unfortunately, is it takes a long time to prove whether your hypothesis is true. But we're not deterred. And the strategy is, in our thinking, is to take samples that were retrospectively collected from patients that had, in the case of prostate cancer, aggressive versus slow-growing cancers. And we're looking for proteases to profile them and see if we can find signatures, make sensors against them, test them in mouse models, and then eventually go back to patients. So I think we are in a time where we have archival tissue from patients that were longitudinally annotated, so we're able to at least do the first part of the experiment. Ultimately, we have to go prospectively, and so that, I think, is daunting.
So I would say, Saengeeta, you know, you've taken one approach which is elegant and has a good chance to work. But here's where I would define the term as translational basic research. This cannot possibly be the only approach to get amplified signals that would increase the sensitivity for detection of early cancer. I mean, that's, without having any evidence for that, just knowing how science is, it cannot possibly be the only approach. And I think the point that Bert made a few minutes ago to the audience, that in today's world, the kind of work that Tyler and I and Nancy and probably others in the audience started in the late 60s or 70s, understand the molecular basis of cancer, and Bert did. That was the exciting frontier. Today, that is largely, I'm sorry for those still doing it, derivative, boring work, because we understand conceptually what's going on in cancer. We really do understand that. Not in every single detail, but we no longer have a conceptual framework for understanding that. The frontier now is to use modern technology, as you alluded to and are developing, to use that information not just to treat late-stage cancers, but to detect cancers at an earlier stage where treatment will be easier and more effective, or prevent them in the first place if there's a special case where that can be done.
So that's certainly true. I certainly agree. I'd have to correct something because you've referenced me in this era in the 1960s when I was in grade school. So can I just be clear on that? So Bert, you are focusing on many things, but certainly circulating cell-free DNA. It has this same challenge. You can find signal. Is it clearly indicative of a lesion that's going to kill you? If not, what do we do about it?
Well, let me address it in a general conceptual format, because I think what Barry said is extremely important, and the challenges he mentioned are real, and they face everyone, including me, who's trying to develop new tests. The word, basically, that Barry said, and is the topic of your question, is this overdiagnosis. Overdiagnosis. I don't like that term. I hate that term. I think there's accurate diagnosis, there's inaccurate diagnosis. And the problem is not with overdiagnosis, it's with overtreatment. The purpose of diagnostic tests is simply to provide information about risk. A test that you've talked about, that I've talked about, only provides information that you are at higher risk, if it's a good test, of developing cancer than you would be if you hadn't been tested. Now, what the patient does with that information is up to him or her. It's not dissimilar to a BRCA test for hereditary BRCA disorder. A woman could elect to get a double mastectomy and a salpingo oophorectomy, but it's up to her, and it depends on many other factors. And I think we need to look at it in that context and apply the same sort of standards to prevention and detection as we do to cancers. For example, suppose one were diagnosed with an advanced polyp. Well, the chance that that will progress to cancer is actually pretty small, but there seems to be no disagreement that the polyp should be removed. You can even extend it further. Suppose it's a stage 1A breast cancer or colon cancer. There's no evidence that taking out that cancer will actually save lives. Maybe all of those cancers are indolent. But we know now what cancer is, like Ed said, and it's progressive. They're going to develop other mutations. It's inevitable. Now, they may not get cancer next year, 2 years, 5 years, but they are at higher risk. So I think we have to apply both intuition and regulatory bars to these decisions, and we will certainly not have all the information we need to base the decisions we make on this information. We're going to have to use common sense and the best collective intuition that we have.
Bill.
So I think that the framing that Barry made is right on. but I'd like to think about it slightly differently. So imagine an extreme example where any intervention is harmless and completely effective. Then you wouldn't really care about about picking up too many whatever if the intervention was completely harmless. So of course that is an ideal and an extreme. But as you move closer and begin to change the equation of harm versus benefit, you could see where you get closer and closer to a time where you will have the rationale to do powerful screening as long as your interventions move closer and closer to be extremely safe. And remarkably enough, the tolerance for things that aren't even that safe, like colonoscopy, are very, very high. But as you got closer and closer to a better risk-benefit profile, the powerful screening tests, I believe, will help save more lives and decrease a lot of the problems that Barry framed up correctly.
But, you know, I'm going to challenge you, Bert, because at least I'm interpreting what you said is that any test that detects anything is a worthwhile test to perform. And then with that information in hand, the patient or perhaps their physician can decide what to do about it. But I'm worried about that. You know, the patient who goes down to the shopping mall and gets a CT scan and finds an incidental lesion in somewhere deep into their body is now likely to act on that, or may act on that. Is that the right thing to do, or do we need more specific tests that will indicate that that's something that we should worry about?
Yeah, no, I'm glad you asked that because it's an important point and requires clarification. In my view, a reasonable criteria for a test is one that has high enough positive predictive value that it's reasonable to recommend. But there are many bars on diagnostic tests, and Barry presented some of them. A bar that says a certain positive predictive value and indicated a certain risk is entirely encouraged, in my view. But the tests in the shopping mall are unlikely to meet that. The problem that some regulators have espoused is that you not only have to show that, but that you have to show it saves lives. Colonoscopy was only shown to save lives 30 years after it was implemented. So I think, and I'd like to hear Ed's perspective on this, and because we were talking about it before, that if you have some reasonably rigorous bars for a test, that's fine. But if you make the bar too high, you'll never get there. And you won't encourage companies, biotechs, to develop the test because they don't have the resources to spend 30 years proving that a test which will obviously save lives will actually save lives.
Yes.
Well, I would say the following. I'll come back to the point I made, but I'll try to be clearer about it. I think that the needs in cancer are different for trying to make a real impact for different cancers. I'll use 2 or 3 examples. I mean, one, take ovarian cancer, pancreatic cancer, maybe uterine cancer as examples. We don't have good early diagnostic tests for those. And when people are diagnosed, they're in real trouble. The treatments aren't great, and they're in real trouble. So I would ask the question in those cases, can we apply the best technologies available and fund research to try to develop early diagnostic tests for those tumors, and then ask the question in trials, as Dr. Kramer articulated, whether that makes a difference to people's lives.
Right.
Now, unless we do that, we're never going to know. So we can argue about whether it will work or it won't work, but I would argue in that case, you know, Doug in his position should influence the funding to develop those kinds of tests in a program to ask whether that can be done. So for something like lung cancer, I think that early diagnostic tests are certainly a way to go, But I think that's really a public health problem, that no one has dealt with in a rational way to make people who decide to smoke, who have the right to decide to smoke, what are the consequences that they need to pay in order to have the right to smoke that does not increase the costs to everyone else in the medical system who doesn't smoke and who are not at the same risk for getting lung cancer that they are because of a habit they've chosen to do.
So that last phrase took us in a slightly different direction, but I want to bring us back to where you put Doug on the spot.
Yeah.
So, Doug, you have the authority, head of the NCI, influence how dollars are spent. Do you want to address Ed's challenge about, and others' challenges about the distribution of research support in this area versus others?
Well, I think that in part because of the funding having been flat for such a long time, all areas of cancer research are underfunded. So the assertion that cancer prevention research is underfunded, true, but I would argue that it's true of our other areas. We are continually struggling with trying to figure out where are the most useful opportunities, and that is not only dependent, the need is there, but it also is dependent on the quality of applications and proposals that come in. We certainly we certainly pay attention to important proposals in the area of cancer prevention and screening because we recognize what critical areas these are. And Tyler is really asking a key question, which is that you really are asking two different issues. One is, do you detect a lesion? But the other is, what's the prognosis of that lesion if it were left alone? And so you're really talking about a risk-based strategy. And in order to do that, I think that two critical issues are, first, to have to have well annotated biospecimens that you can do analyses on, and second, to have more information about the natural history of different kinds of tumors with a variety of molecular abnormalities. Yes. And almost certainly the importance of the microenvironment is only going to become greater as we have more understanding of cancer.
Bert, I'll give you the last word here, and then I'm going to switch topics slightly.
Okay, so this addresses the question of bars.
Need your microphone, Bert.
Oh, I'm sorry. This addresses the question of bars and the difference. First of all, I sympathize with Doug. You can't change what's going on in Hait. It's this huge ship that's going its own direction. But at any rate, there is a difference in the way treatment for advanced disease and early detection and prevention are handled, and it's pervasive. And I want to ask both Bill and Heidi if they could perhaps comment on this. Let me give you an example. Bill, if your bar is to develop a drug that cures cancers and causes minimal side effects, you might as well go out of business, right? So it all depends on what the regulatory bars are for approval, whether it's a test or a drug. Second of all, our economic bars for tests, and this is where Heidi gets in, are much different than for drugs. Economists figure out whether a test is useful. It depends on how much it costs per life saved. And if it costs more than $10,000 or $20,000, it depends on the economist, et cetera, no good. However, when we talk about treatments, never an issue. And I think a guy named Tom Foreman wrote an editorial about this in New England Journal a couple of months ago. It costs $1 million, millions, and it does for treatment. But nobody ever asks how many years per life saved or per month prolonged. How do we get that kind of perspective, Heidi, from an economist's view into the public arena?
So I will say that that's actually more of a public health perspective than an economic perspective. I would say is sort of thinking about the cost per life saved. One way that I would think about this is to go back to the HPV example. So a thought experiment you can run in your head is, would the HPV vaccine have gotten developed if we would have required it to show that cervical cancer incidence declined, as opposed to that HPV incidence declined? And that's sort of a question that we'll never know the answer to. But people have speculated that actually using HPV as an endpoint in the clinical trial was really important in bringing private research dollars into developing that vaccine. And you can look at other historical case studies for drugs to reduce blood pressure or reduce cholesterol, rather than showing that the drugs actually reduce cardiovascular disease, as another example where people would say that those endpoints, which are often referred to as surrogate endpoints, were very important in spurring the development of some drugs that we now think of as socially valuable. And so one way of thinking about innovation in this space is what standard we use for evidence is partly a function of the scientific basis for potentially having outcomes that we can observe on a shorter time horizon that are predictive of the final outcomes that we care about. And so that's sort of an area that I think is very ripe for public investment, because one private firm that has an idea for how to come in and develop a new diagnostic test, it's not in their interest to come up with a marker that's going to allow every other diagnostic company to show that their diagnostic can also get approved. That's a natural place where economists would say there's a public good problem where the NCI or other people should come in and subsidize the development of these intermediate endpoints that may serve as markers that people's health outcomes are going to improve later, as a way of leveraging private investments. And so I think rather than thinking about this as sort of what's the bar from a cost per life save perspective, thinking seriously about what's the role for public investment in sort of developing new bars that can actually shorten the time lags and shorten reduce the costs, of developing new diagnostic tests and also developing new treatments, I think is sort of a very concrete way in which public policy could potentially have an important impact on this space.
So you will respond,
yeah.
and I'm also going to ask you another question, because you're going to be put on the spot twice here. So I'm interested in your response. I completely agree with you, Heidi, and you're sitting in front of a room full of people who can help provide those surrogate markers to help accelerate and prove the value of the tests that are eventually developed. So Bill, I'm happy to have you respond to Heidi, and then I'm also going to ask you a question.
Okay.
Because Bill, you may or may not know, at Janssen, part of Johnson & Johnson, has been very public about his interest in advancing an approach that he calls disease interception, and I'm not putting words in your mouth, and that is to do something that Bert actually described earlier: find early disease and try to stop it in its tracks. Okay. And that's not an approach that industry has adopted widely. I think you would agree. So I'd like you to talk about that. Sure. And while you do that, I'm going to grab the questions from the audience. So okay. You're on.
First, let me start with where Bert was going, with where the bar is set. And in fact, if you really step back, the incentives for us in the pharmaceutical industry are actually quite perverse. Because where we start, let's say in oncology, we take the most advanced patients, and then all we have to do is show some incremental benefit in progression-free or overall survival in patients who are far down the line, who actually benefit the least from the therapy, and usually suffer the most side effects. And that can get you, in the US anyway, an approved drug and on the market at an extraordinary price. I shouldn't say anything about price. I'm in the pharmaceutical industry now. As you move earlier and earlier in the disease, the trials get longer and longer because the event rate in the control arm is slower and slower. So when you get to the area of people who have pre-malignancies or prevention, if the same endpoint, as Heidi was pointing out, would be something way downstream, the incentives, the market incentives for a company are very, very limited. So much will rely on surrogate endpoints, and of course Merck was the leader when, in a different era, perhaps in a bygone era, when lowering cholesterol was a good enough surrogate with enough data that today probably wouldn't be enough to get the drug approved, and of course the results have been miraculous. In terms of what we're thinking about at J&J is the following. we ask, well, what do patients want? And as a medical oncologist who practiced almost 30 years, no patient ever walked into the clinic and said to me, you know, Dr. Hait, I'm so happy that I've been diagnosed with cancer so you can treat me. Haven't heard it yet. You know, someday maybe. I don't think I will. But rather they say, how did I get this disease? I never smoked, I never drank, I'm a vegetarian, I lead a clean life, I exercise regularly. Could I have done something to prevent the disease? If I knew I was on my way to getting something, if I had an in situ problem, could I have made an interception of the process? Or now that I have the disease, can I be cured? And if you look at the way we focus on things in oncology, not in infectious diseases, but in oncology, we don't set out to cure the disease in the drug development. We would like to, but that's usually the way the process works. It's not about curing, because that would take combinations and you'd have to control it perhaps. Immuno-oncology might be the exception. In terms of intercepting the disease, we don't know enough about the pre-malignancies. I mean, the one area that was most shocking when Bert really described for us many years ago the progression of a normal colonic or an at-risk colonic epithelium through a polyp to disease, you would think there'd be thousands and thousands of sequences of every omic imaginable on that progression pathway in publicly available databases. It's remarkable how little knowledge has been accumulated in that space. And prevention, again, it's a knowledge problem in oncology. We don't have enough knowledge to really know, with the exception perhaps of some of the infectious causes, how we prevent these diseases. And unless there's a change in the ability to accept surrogate endpoints, it's very hard to imagine a market for how companies will step in and be willing to invest the sometimes billions of dollars to get there.
Ed.
I want to pick up on Heidi's comment, which is really important, and I want to continue to tweak my friend Doug Lowy, who I've known for a really long time, so it's all in good sport. Investing in epidemiology and natural history of disease with modern technology as opposed to classical observations that are made clinically in epidemiology is absolutely a critical area of research for then helping companies decide where they're going to invest. Because without that information, it's extremely difficult to make choices. And Doug, I would say, you know, companies never have enough resources to work on everything, and they're forced by the nature of private enterprise to make choices about where they put their most of their dollars. They make bets for what's going to be the largest payoffs. And I think that public institutions like the NIH and like the Cancer Institute will never have as much money as they need or as they want or as investigators want. And it's really up to the leadership of those institutions to make, again, the best choices they can make with the given information that's available at a given point in time, where they think they'll have the biggest impact from the public research that they're funding. The country has a $17, $20 trillion debt. I don't think we're going to see the kind of massive increases in public funding for research that we'd all like to see in that climate. And so making difficult choices where you think you'll have the biggest impact on public health is really going to be a critical component of the leadership of the various institutions at NIH.
Doug, would you like to respond? I'm not sure there was a question there. It was a little mini lecture maybe.
It was a lecture. Yeah. I said he's my friend. We've known each other for 40 years.
I certainly think that when you consider the investment, if you will, basic science, prevention, screening, treatment, on the treatment side, private philanthropy is very oriented towards translational and clinical treatment research, and the vast majority of private funding for profit research. Bill is not the only outlier, but certainly what Bill tries to do in the areas of pre-cancer not shared by most private industry. And so what the NCI does is really to add value in the area of treatment to the much larger investments of private philanthropy as well as in the private sector for treatment. But when you go to the more upstream areas of basic research, prevention, screening, and population-based research, it's a slight exaggeration to say we are the only game in town, but not a tremendous exaggeration, and I am explicitly aware of that.
Yeah, no, I agree. That's why I went off on my soapbox.
All right, so I'm going to spend 5 minutes asking just a few questions, and I apologize. I want to give you 75 minutes for lunch. So, and there were many very good questions, and thank you for that. I want to first turn to the question of diagnostics again, and several actually asked about, why is it so expensive? What are the limitations? Why is the FDA a problem? Why do they get reimbursed so poorly? So we touched on that earlier, but I think we could talk a bit more about it. And Bert, you might want to tell the story of Exact Biosciences and their colorectal test to emphasize some of the challenges. And Heidi, I would invite you to weigh in as well.
Sure. Exact was a fantastic experience, a long story, which I'll make short. But basically, David Zadransky, when he was in our lab in the early 90s, published a paper in Science showing that you could detect mutant DNA, mutant KRAS, in the stool of colorectal cancer patients. I remember being asked a question in a lecture, well, how much did it cost to do this per patient? And the answer was about $20,000 in $1992, so clearly wasn't ready for prime time. But an engineer, an entrepreneur named Stan Lapidus, came to us several years later, asked if he could license the technology, and started this company called Exact. Make a long story short, the company had a lot of problems, almost failed, but at the end developed a test. This is the first test. It's called Cologuard. That's based on this concept of released tumor DNA, because they detect DNA in the stool as one of the components of the test. It took 60,000 pages of documentation to convince the FDA to approve it. 10,000 patients. Barry Berger, who lives in Boston, and I think at least some of you know him, was instrumental in that approval. But that was only one of the hurdles. There are other hurdles too. You have to convince the insurance companies, the health maintenance organizations, Medicare, that it's worth paying for. All challenges, but now all overcome. It is possible to do it. Furthermore, and perhaps more importantly, independent analysts suggest that 40 million people are going to avail themselves of this test over the next decade. So it's real. The test isn't perfect, but I think one of the important lessons from that story is, first, it's possible with enough effort, and second is, once you get a test, it's much easier to get a better test through the FDA, because then you don't necessarily have to show it saves lives. You just have to show it's better or equal to the first test. Right. And that's a much lower bar. That's why I think for all of these early diagnostic tests, the first one, proteomics, DNA, nanoparticles, I don't care, that gets approved and has a high enough positive predictive value, et cetera, will make it easier for all subsequent tests. And I think Exact really should be congratulated for what they did. It wasn't easy.
Any other comments along those lines? We've just got a few minutes left. Another set of questions related to sort of the cost of implementation and some of the sort of global health challenges. We focus on our own problems, but we recognize the problems are global. Saengeeta, you discussed a bit about your goals downstream of dealing with this issue. Do you want to say more? Others want to say more about how we're going to sort of democratize these technologies to make them affordable to the world?
Sure. I think one thing, engineers like to solve problems, and I think if you look at global oncology from sort of an engineering lens, you might ask the question differently for early detection than the way it's playing out in our current kind of research climate. So for example, HPV has been an amazing vaccine, but there are places where it's still not licensed. Here in the US, we think cervical cancer screening is a solved problem, right? But there are places in the world where it does not exist. There is no cervical exam, there is no pathologist, right? And so, and that drives the numbers that Doug showed on his slide. And so if you say, okay, this is the reality, then maybe you would say, how else can I, now we know HPV is the causative agent, can I make a urine test for HPV? I know which are the bad strains. You might reinvent a completely different approach than the one that the problem already looks solved. And so I think, for the trainees out there, I think I don't know enough about the global oncology environment, but I think it would be worth us learning more so that we can kind of revisit some of these questions with what is now known.
Doug.
Well, like all of cancer, these are multifactorial issues. But if you don't do anything, you're not going to make progress. And so you need to decide what area or areas will you work on. And in the global arena, one of the reasons why people focus on cervical cancer is, first, it is a big problem. The chance of a woman in the developing world dying of cervical cancer before she's 75 is 1%. But there are two modalities, both of which are prevention-oriented. It is far more complicated to do screening than it is to do vaccination, especially if you get down to one dose. Every 11-year-old girl, when she's 11, you get vaccinated. But it's going to take a good 30 years before you see an impact of that, because cervical cancer doesn't develop until you are in your late 20s or early 30s. For women who are at risk of developing cervical cancer, the key in the developing world is to figure out how to do screening while at the same time not need to build up the extraordinary infrastructure that we have in the industrialized world. And my own view is this is where technology will be able to replace the pathologists, the gynecologists, the cytologists, et cetera.
I agree. I think that's an engineering challenge.
That's what I'm saying.
Yeah.
It's technological innovation that can replace the expertise that we have built up over many years, and be able to go in and have an impact that way.
All right, I'm going to ask one last question, and then I'll let us go to lunch. And I didn't prepare you for this question. It's going to be a hard one for you to answer. You alluded to it, Ed. We are focused today, all of the talks are focused on science and technology and how we're going to bring that to the problem of early detection and prevention. When hanging out there as the big elephant in the room are the lifestyle changes that would be necessary to make an even bigger impact than everything we're talking about. Smoking, 150,000 lives saved each year. Obesity, and many other risk factors that we know about today. So how, as a society, are we going to make progress in those areas? Not necessarily the expertise of anybody on this panel, with the possible exception of Doug, because he has to think about this stuff. But how are we going to make those changes? Just in a word, and then we'll close. And I'll go down the line.
So I think what Nancy brought it up in her introductory remarks, getting the best and the brightest, or more of the best and the brightest, to focus on behavioral science, or whatever the proper term of that area would be remarkably important. And the one example, we're thinking about, okay, how do we prevent or intercept lung cancer? And you say, well, what causes lung cancer? People say cigarette smoking. And then you say, well, what causes cigarette smoking? And there's silence. And I think that huge gap in knowledge is what we have to address.
Heidi.
So this is a very specific example, but in general, I think it's worth more people spending time thinking about who has the incentives to improve behavior, and is there a way that we can leverage that in a way that really would have positive effects on population health. And so the specific example was from our department faculty lunch this week. We were noting that a lot of universities have moved to subsidizing people to use the gym, because universities are traditionally self-insured, which means that if their health costs are lower, that benefits the university. And MIT is a real outlier there, where for both faculty and students, the cost of going to the gym is actually pretty high. And so I think thinking about things that employers could do as one example of an institution that has the incentives to reduce healthcare costs as a side effect would also improve the health of the population, and using that as a lever for improving population health and population health behaviors, I think is sort of a thing that's worth thinking more about.
I'm going to be repeating what people said. Research on behavior and incentivizing healthy behavior.
Bert.
You ask a bunch of scientists about how to solve a problem, and they're all going to say more research, right? So that's what we're all saying. But we're also saying that Doug should devote $100 million next year to that behavioral research, and that would likely be one of the best investments you could make.
Saengeeta.
So I think this will, what I was saying earlier, I want to just underscore, which is I think we need new minds to tackle some of these things. I think incentives is a really interesting idea. Engineers, we have wearables. There's a lot of technology now that we didn't have when we thought about this last time. And so I really like the idea of sort of some big partner and an interdisciplinary team and making them bet on the colon cancer screening test before we know it's going to save lives. I think we need some bold leadership and a lot of new ideas. And also, I think to convince the talent that this is I hate to use this word, but it's like a sexy area. This is exciting.
Right.
I think it's not just, it's not, cancer biology research is really important, but this is a piece of it that is, hugely impactful.
Yeah. Ed,
I think
word.
you need both positive incentives and negative incentives to get people to change their behavior. And I think that's a very, as you point out, it's a very complicated problem. And I think the public health system in this country, perhaps in all countries, has really not dealt with that for the major causes of cancer, and that they really should. And that is a role the government should take leadership in.
Good. I'm going to just quote Barry Kramer, whom I talked to earlier, more or less about this issue, and he said, it is a perception problem, like you were saying, Saengeeta. It's a bit of a perception problem. He said, the heroes are the ones who cure the disease. They're the heroes. But if you could prevent even more disease, you should be a hero too. So I think we do have to change the perception here. All right, thank you to the panel. Please join me.